Hello, all. Um, we're going to talk about 6.36, which is asking us to tell the difference, right? Tell the difference between something called an antigen and something called an antibody. So that's what we're going to do here. Before we do, I just want to review real quick some of the concepts earlier that we talked about. So earlier in this topic, we talked about, let me make that a little. We talked about non-specific immunity, and then we talked about specific immunity. So let's just talk about the differences between these two. I'm going to write right over the objective, but oh well. So non-specific immunity is when your body responds to something that not specifically. So let me clarify that a little bit. So it could be anything. So let's say your body is trying to defend you against viruses. So that is a very bad drawing of a virus. Kind of looks like a turtle. Maybe your body is trying to protect you against bacteria. Maybe a parasitic worm. So your body has all these different things in place to protect you from this. But it doesn't matter which one it is. We're just saying we're not specifically targeting a certain type of pathogen. Remember, pathogen is anything that causes a disease. But we're just trying to keep everybody out. So the main way your body does that is your skin. So the outside of your body is protected by this amazing organ that doesn't let any of these guys come into your body. However, you've got some holes in your body. So specifically on your face, your eyes, your nose, and your mouth, there's ways that they can get in. So there's a second type of nonspecific where you've got these cool white blood cells that patrol your body and they consume these pathogens. They engulf them. It depends on how big it is. It would be hard for them to consume a worm, but bacteria and viruses for sure. They consume them and digest them and then release their contents after they've been digested. So those are two non-specific ways. Specific immunity is, just as the name suggests, is that we're identifying a specific pathogen. And the way you identify the specific specific pathogen is by identifying something called an antigen. So on the surface of a bacterium, on the surface of a virus, there are proteins. So that's what these antigens are. Now I know in the definition I have here it says substance, but for now I just want us to think of them as proteins. So just like on the surface of your cells, you have surface membrane proteins, right? Well, so do bacteria. And also, so do viruses. So I'm going to draw some antigens on the surface of this. I'm going to make them green for whatever reason. So here are some antigens on the surface of this bacterium. The important thing to understand about these proteins is that they are considered not self, which means they are not you. So this bacteria is roaming around and your body has some steps in place to be like, hey, wait a minute, that protein isn't ours, so we are going to do something to get rid of you. And how do they get rid of you? By identifying your antigens, by identifying these non-self proteins. They produce antibodies. So antibodies, they are you. They are produced by you. They're actually produced by white blood cells, and we're going to talk about the whole process involved in activating this response. But they're produced by white blood cells called B cells. Very, very important cells in your body. So somehow this B cell gets information, and we'll talk about those techniques, and about this non-self protein, about this antigen, and it's going to produce an antibody. So antibodies typically are drawn, like in this picture, as these Y shapes with little binding sites on the outside. Now, this is not drawn to scale, of course, but a B cell will produce all of these. These proteins, they're globular proteins. B cells will produce many of these proteins, especially when you have a very large infection. And what happens is that these binding sites will attach to these antigens on the surface of the bacteria or on the surface of a virus. And when they attach to it, three things can happen. So one thing that can happen is that they make it hard for the bacteria or the virus to do anything. Like if you remember, bacteria have these surface pili on the outside that allow it to stick to surfaces. Viruses have proteins on the outside that allow it to enter your cell. So by sticking to the outside of it, they can't do their job. Number two, you can make other bacteria stick together. So if I have another bacterium here, and we haven't, actually I should have drawn it over here. So let's say we have another bacterium over here, and it has an antigen on the surface over here. Now I've stuck two bacteria together, and they're just going to be clumped, stuck together. So it's going to be easier for these guys to come around and munch on the bacteria and eat many of them at the same time. And the third thing they do is they kind of like have a little flag on the surface to help identify. So by having your antibody stuck here, it identifies it to your macrophages, those leukocytes, to identify them and be like, hey, over here, come eat him. So those are the three ways that those work. So in summary, real big, antigens are not you. Antibodies are produced by you. Antigens are proteins on the surface of these pathogens. Antibodies are globular proteins that will attach to these antigens in response to your immune defense system. So that is it for this. I hope that was clear enough. Thank you.